les discussions quand même qui sont très intéressantes et les questions. Donc, Anthony, si tu veux continuer à répondre aux questions aussi sur le chat, n'hésite pas. OK, so, le prochain talk, Pierre-Nicolas, je te laisse.
Oui. Yes, so the next talk will be by Laurent Bréhélin from University Montpellier, and work that was done during the PhD of Christophe Menikeli. So please, Laurent.
Yes, thank you. Thank you very much, and thank you for this invitation. That gives us the opportunity to present some work that we have done in our group here in Montpellier. So it's an inter-laboratory group that we created, I think, 6 years ago now with Charles Cellier and Sophie Lebrun. So as you know, gene expression is regulated in eukaryotes at different levels. There is transcription level, there is a post-transcriptional level, and among all mechanisms that are at the transcription level, transcription factors are supposed to play a key role, yeah, by binding the DNA at precise locations and recognizing specific motifs. And in previous works, we have proposed methods that predict whether a sequence is bound or not by a specific transcription factor. This approach is based on a logistic model that takes into account the motif of the target transcription factor, but also all the motif of the other transcription factors that are the cofactors of the target TF. That is all the TFs that co-bind around the target TF. And this approach is is a logistic regression model that is penalized with a lasso penalty, which is a well-known statistical technique for penalization of the likelihood, that has a great advantage to do feature selection, which means that in our case, we can train a predictor, and at the same time, we can learn which are the cofactors of the specific TF, of the target TF. And for those interested, you can have a talk tomorrow by Rafael Romero, who is a PhD student in the group, that have greatly improved this approach, and that use it to analyze the genomic features that are involved in the cellular specificities of transcription factor binding. So don't miss this talk. I think it is perfectly in the topic of this symposium. Yeah. So today I would like to speak about another kind of motif that we call long regulatory elements, and that are longer regions that span several dozens or several hundreds base pairs that have not clear motif, but that are enriched for a specific nucleotide or specific chimer, and that are also linked to gene expressions. So the most famous example of these long regulatory elements are, of course, the CPG islands in vertebrates. But there are also other examples. For example, the GIMREC lab that shows that the short tandem repeats that are long repeats of very short chimer that are 1 base pair or until 6 base pairs. So this very long repeats also are involved in the gene expression regulations. Yeah, there are also the example of AU-rich elements in 3' UTRs. And so some authors have proposed that this domain with specific base compositions may actually mold the epigenome. And in line with these points, in a previous work, we have shown that actually we can predict the gene expression in human simply using the dinucleotide composition of specific parts of the gene. So what we did in this paper is that we compute the frequency of all the dinucleotides in different regions. So we compute the dinucleotide frequency in the core promoter, in distant promoter, in the UTR regions, in intron, in CDS, and so on. And we use all these dinucleotide frequency computed in all these regions to build predictors that predict the gene expression of the genes. So we have in this works 160 variables, and we learn linear predictors that predict gene expression from these variables. And we did this on TCGA database, on patients from the TCGA database. And we use a model that is learned for each patient to compute, to predict the expression of the genes of the patients. And when we compute the correlation between the predicted and the measured expression, and we show that actually we can predict with moderate, but I think surprising accuracy, the gene expression using only these dinucleotide frequencies. And another thing that we show is that, of course, CpG islands in promoters are very important features to predict the expression, but it is not the only features. And there are also other features that are not CpG islands that are really helpful to predict the gene expression. So in summary, the expression, at least in human, is linked to dinucleotide frequency of long regions. And we wanted to know if we could propose a method that can identify the region automatically. We ask if maybe there is something above the dinucleotide level. I mean, maybe 3-mer and 4-mer are also informative of gene expressions. And we would like to apply this kind of analysis on very different species. And I think that there are a lot of bioinformatics tools to discover motif. I think that motif discovery is one of the oldest problem in bioinformatics, that there is no bioinformatic tools that are specially dedicated to the identification of these long regulatory elements. So we proposed for this the Dexter approach, which take in input an RNA-seq data and a sequence for each gene. So for each gene, we have a measure of gene expression, and we have a sequence, and the sequence is aligned on the TSS of the genes, and the sequence span 4 kb base pairs. And what does Dexter do is that it tries first to identify a set of variables that are correlated with the expressions. These variables are actually a pair of a K-mer and the specific regions in the genes. And then once you have all these variables, he builds linear models that predicts the gene expression from these variables. So for the first step, the step of the construction of the variables, what Dexter does is that he takes one dinucleotide, okay, and he builds this kind of structure, which is a lattice actually. So you have the sequence here, you have the TSS here at position 0, and each node of the lattice actually corresponds to a region in the sequence. Okay, so this point corresponds to this region, the top of the lattice corresponds to the whole sequence. And then Dexter computes the frequency of the dinucleotide in the specific regions, and he does this for all the genes, and then he computes the correlation between this frequency and the gene expression. Okay, so I have an example here. It's the lattice for the dinucleotide 80 in gene expressions that come from the P. falciparum, which is a parasite responsible for the malaria. And what you can see here is that the frequency of the dinucleotide 80 in these specific regions here, the specific region is a region in between -1 and -1241. Okay, the TSS is here at position 0. Then if you compute the frequency of 80 in this specific region and you compute the correlation between this frequency and the gene expression, you get a correlation of 43. Okay, while if you do the same thing in these regions here, which is a region in between 0 and 1241, you get only a correlation of 4%. So this means that we have huge differences on correlations for the same nucleotide, depending on where you compute the frequency, in which region you compute the frequency. So once Dexter has identified the region with the best correlation for one dinucleotide, it tries to extend the dinucleotide. So we get this kind of exploration graph where you have, for example, here, the correlation between the frequency of TI in the wall regions, which is 26%, excuse me. While when you compute this correlation in this region, the correlation go up to 43%. And then if you increase TI with a T, so you compute the frequency of TAT here, okay, in the same regions, then the correlation still increased to 47%. So at the end of the process, Dexter has enumerated and found out lots of new variables, these variables being K-mer associated with the specific regions. It's mainly two-mer or three-mer, okay? And in the second step, we want to train linear models that will predict the gene expression on the basis of all these variables. And here again, we use the lasso penalty in order to have feature selection and to select the best variable, is the most important variables to predict the expressions. So we did this on different eukaryotes, on different gene expression series of eukaryotes. We did this for Plasmodium falciparum, the agent of human malaria. We did this for another Plasmodium berke, for Toxoplasma gondii, for the yeast. And we did this also on different series of that measure gene expression in different tissue or that measure gene expression along the development for human here, for Arabidopsis thaliana here, for the Drosophila here, and for C. elegans here. So in each series, what we do is that we take one condition, we run Dexter, and we learn a model that predicts the gene expression of this condition. Okay, and the gray bar that we can see here measures the correlation between the prediction and the real expression. And so what you can see in these wall pictures is that this correlation is globally, generally, in between 50% and 65%, except for the Plasmodium species, where this correlation is above 70% for several of the condition years, which was a first interesting point, because actually these species, Plasmodium falciparum especially, are known to have a very few transcription factors, at least non-transcription factors according to the size of the genomes. So now what we wanted to see was if the same model was learned at each condition of the same series. So we did a kind of swap experiment for this. So for example, if I zoom in this, you can see that the model that has been learned here at 0 hour of this life cycle of Plasmodium falciparum has an accuracy of 73% at this time point. But we can apply this model learned here to predict the gene expression of the other time point, okay? And you can have, here you can see the red curve here that show the accuracy of this model on the other time point. And we see that actually the accuracy of this model go down when you, for the late time point, okay? It's not as accurate in the late time point that it is on the first time point. And that is true also for Plasmodium berke. Actually, the effect is even stronger. For example, the accuracy of the model that has been learned on this specific stage that is called the trophozoite stage has an accuracy of 70%. And if you look at the yellow curve here, you can see that this model has an accuracy on this stage here, the gamma histocyte stage, of only 30%. while the model learned at this stage here has an accuracy of 60%. Okay, this means that it is not the same model that is learned at the different phase of the life cycle of Plasmodium berghei or of Plasmodium falciparum. And this means that it is not the same long regulatory elements that are used at the beginning of the life cycle and the end of the life cycle. Okay, what is striking, however, is that when we do this swap experiment on tissue, we have very different results. For example, for human, we have this. It's approximately the same model that is learned in the different tissue of human, and that's true also, for example, for Drosophila. Okay, it's approximately the same model that is learned in thorax, for head, or for abdomen. Okay, they seem completely interchangeable. But another interesting point is that when you look at the Drosophila melanogaster development, you see other things. You see that you get this dynamic behavior, that is the model that is learned in the early time point of the development of the Drosophila melanogaster is not a good model to predict the gene expression at the end of the development of the Drosophila melanogaster. Okay. So now an interesting question that be raised by one of the referees was the link between long regulatory elements and the genome AT content. Because that's true that the species for which we got the best accuracy, the highest accuracy, were for the species with high AT content. So that was the two Plasmodium species. So what we did is to take two other organisms that are not Plasmodium, but that also have a high AT content in their genomes. It's Dictostelium discoideum and Tetrahymena thermophila, and we run Dexter on this species on two different series of gene expression, and we got a very different results. For Dictostelium discoideum, the accuracy of Dexter is quite high, is around 70%, and we got this dynamic behavior. While for Tetrahymena thermophila, the accuracy is around 50% only, and we don't have this dynamic behavior. It's the same model that is learned at the different phase. Now, another question that we were interested about is where these long regulatory elements are involved. Are they involved in transcriptional, or are they involved in post-transcriptional regulation mechanisms? So to respond to answer the question, we use a gene expression series for in Plasmodium falciparum. It's the gene expression of Painter et al. That have measured independently the level of nascent transcription and the level of stabilized mRNA along the life cycle of Plasmodium falciparum. So here again, we run Dexter on this gene expression series, and we identified all the most important features, so the most important long regulatory elements. And we did two groups. The first group here is the features that are more correlated with nascent transcriptions and less correlated with stabilized mRNA. Okay, you have here the features, and you have here, for example, the correlation at with the nascent transcription at 0 hour. Here is a correlation with the stabilized mRNA at 0 hour, and you have this for 8 hours, 40 hours, and so on. So all these variables here are more correlated with nascent transcriptions here that with stabilized mRNA here. So the correlation may be positive or negative, but it is always more correlated with nascent transcription. And here you have the features, the long regulatory elements that are more correlated with stabilized mRNA than nascent transcription. And what is striking here is that all these variables are associated with upstream regions, while all these variables here are associated with downstream regions that are downstream the TSS. And another thing was that these variables are not strand specific. That is, if you measure the correlation between TG, for example, here in this region, in the positive strand and in the minus strand, you have the same correlation with expression. Okay, while these variables here are really strand specific. So it seems that at least for Plasmodium falciparum, these long regulatory elements are associated both with transcriptional mechanism and post-transcriptional mechanisms. And now to finish, we wanted to check whether these variables are really important and to assess their importance by an experimental validation. So what we do is that we take the most important variables, the most important long regulatory elements in Plasmodium falciparum. So you can see here that, for example, this variable, which is the frequency of ATA in this specific region and upstream the TSS, is highly correlated with gene expression, especially in the first time point of the cycle. Okay, so what we did is that we built two chimeric promoters. The first promoters contain 3 repetitions of a short sequence with very high ATA content. Okay, while the second promoter contains only 1 repetition of this high ATA sequence, and the remaining of the promoter has a low ATA content. Okay, and these 2 promoters were integrated with a GFP gene into the same locus in Plasmodium falciparum using CRISPR-Cas9 technology, and we measure the transcriptional activity of the 2 constructs, and we observe that actually the activity of the promoters with 3 repetitions, so a promoter with very high ATA content, is 10 times higher than that of the promoter with only 1 repetition. Yeah. So in conclusion, I hope I have convinced you that long regulatory element may be important mechanism and important elements for the regulation of expression in eukaryote. Their importance, both in terms of accuracy of behavior that is dynamic or static, depends on the species and then conditions, which we observe that it may be more dynamic in developmental series that when you look at different tissues. At least for Plasmodium falciparum, these elements are associated both with transcriptional and post-transcriptional mechanisms. And of course, the big question, and I have not the answer, is that how do they work? I don't know. They may be loose binding sites for specific transcription factors or for specific RNA binding proteins. They can be linked to DNA shapes. They can drive epigenetic modifications. They can have a control on nucleosome occupation. And also another question is why are they so important in Plasmodium falciparum? Is this really linked to the paucity of transcription factors in this species? It's also an open question. And I would like to finish by thanking my co-author. So Christophe Medichelli is a PhD student of this study, Charles Le Cellier, Sophie Lebre, and many thanks also to JJ Lopez-Rodriguez lab. JJ is, I think, the specialist of CRISPR-Cas9 technology for Plasmodium falciparum. And thank you to listen to me. I'm done.
Thank you, Laurent. Do we have questions? No questions among the participants?
I do have a question, if I may.
Yes, yeah.
Yeah. A couple of ones, actually. So the first one is, did you see any relationship with the size of the genome? Because it looks like you can extend like 2 kb upstream of the TSS, and in a genome like Plasmodium falciparum, where you have way smaller intergenic regions, then you might start to already get some information from the gene that is nearby, which you would less likely have in humans, for instance. And how much would that impact the results? Because if you're learning information about the codon composition of the nearby gene, then you might learn information what are the codon biases that are used, which we know that genes with the same codon bias tend to be expressed together?
Okay, I don't think it's the case, but where you are true is that in Plasmodium falciparum, there is very few an answer. So I think that one of the reasons why it works best in Plasmodium falciparum is that actually all the code is in the promoter and near the TSS. And so it's simplest to predict expression in this condition, I think. But I don't think it has a link with the previous gene. Actually, when we do the CRISPR-Cas9 test, we have a look at this question carefully, and the promoter that we construct was we take great attention to do not incorporate in these promoters a neighboring gene that could be here. So I don't think it's the case here.
So that would be interesting to remove the exonic regions from the regions that you learned, so that you would make sure that you only have regulatory regions when you train your model.
Yes, but this genomic region could be also regulatory regions.
Well, it's really rare. Having an exon that serves as a regulatory element, it's almost never been seen.
It must be proved, but I don't think it's so weird. It's close in the genome.
Yeah. Yeah. Okay. Thanks.
So we have a question from Raphael Mourad. So if I understand well, you assume a somewhat constant distance between the TSS and the long regulatory element. So do you plan to apply this constraint?
Yes, that's true. That to apply this approach, we need to have an anchor, kind of anchor to align the sequences. And we actually are working on a method that use actually hidden Markov models and that do not have this constraint. So we could, with this approach, scan all the sequences without having all the sequences aligned on a given point.
Okay, thank you. We have a question from Pascal Martin.
Yes.
So about which
yes,
so in which tissue you have the highest correlation?
Yes, that's true. If I share my screen. That's true here. That for Drosophila and for C. elegans, we have higher correlation on gonad, and actually this tissue seems a little bit different of the other tissues. And what I said earlier is not exactly true for this tissue. So the model learned in this tissue are not as good for gonad than as the model learned directly for gonad. But I don't know why. I suppose that it is related to the development also. But I don't know exactly why. No.
Thank you. So if we don't have more questions, thanks again, Laurent.